Hello, my name is Benjamin Aaronson. I am a senior biology and Spanish major at UMass Amherst. I am a member of the 9th cohort of UMass Amherst's ICONS program, and I am on the biomedicine track. I'd like to warmly welcome you to my TED Inspired talk, and thank you so much for listening. I'm a member of the Albertson Lab, operated by Principal Investigator Craig Albertson. We study evolution and development using fishes as a model organism. A lot of my work has been focused on the jaws of cichlid fishes and how they are responsive to the environment. So, why are you here? There is a problem that is occurring now and will keep on occurring in the future. You are interested in a possible solution for this problem. So, what is the problem? Bone disorders, particularly those involved in the developmental stages of children. Common disorders include polydactyly, syndactyly, radial hypoplasia, and frontal bossing. Most of you in the audience listening to this video currently suffer from one of these disorders or know someone who does. What you see is an example of frontal bossing in a young male child. This is radial hypoplasia. You may be wondering what the prevalence of these disorders are, and it is about 1 in 1,000 births. In the US, to put it in a little context for you, there are approximately 10,000 babies born every day, and that means roughly 10 babies are born with one of these disorders or one like it each day. This is syndactyly, and this is polydactyly. In every case, some malfunction or some abnormality in a person's bone is observed. So why is this a problem? And these disorders are a problem due to the difficulty they cause during the physical development of children. And here we're going to hear a little bit from Dr. Amy Ladd, who explains bone disorders really well.
So, Walt Disney made, back in the day, hand drawings for cartoons. There were every frame was an individual hand-drawn picture, and so he would do the storyboard, so the big pictures, and then in between, these artists called tweeners, or the in-betweeners would do the drawings, the separate drawings, and you have to link them together. And he made it the standard because it was 20% fewer fingers to draw. So in homage to Walt Disney, every cartoon character that has kind of a human hand look has 3 fingers and a thumb. And so who needs a full 4-fingered hand? So here's this kid 10 years later, and he's driving, so he's doing those tasks again. And I'll show this to medical students to say, what's wrong with his hand? They usually say like scars or something. It takes them a while to figure out that he doesn't have a finger. So this awesome kid finds me 10 years later on LinkedIn, and I asked him if he will reproduce the test that he did when he was 18 and 6. And so he's showing you these. So the left hand is the one that did not policize, did not do too much in the way of fancy stuff. And so that's his left hand, functions very well. And then the right hand, the one that you just saw when he was first driving. I love that little trick. I can't do that. And he went to the next step. So it's going to write his name. It's not totally normal, but works really well. And it's a cool procedure. So then he put his GoPro on and shows how it is. And certainly faster than me because I have no idea how to do any of that stuff. But so LinkedIn digital age, pretty cool.
So I'd like to thank Dr. Amy Ladd and give all credit where credit is due. But the point of the story is that with fused digits or extra digits, polydactyly or syndactyly, or bone disorders pose a great challenge when children are learning to walk, run, maintain balance, learn how to write their name, etc. Frontal bossing can cause facial bones to be fragile during development, as well as cause some bones to not develop at all. So you may be wondering what some causes of these disorders are, and major causes include genetic factors. So using Play-Doh, I'm going to show you how developing bones are how malleable developing bones are, and how easy it is to bend and cause an abnormality, right? With Play-Doh, it's really the same going on with developing bones. It's easy to have fused bones or get an extra digit out of nowhere, right? And like with certain developmental cues, can throw a wrench into how bone formation is occurring. Other causes are environmental causes. So being a diabetic or having a premature birth, if you are an epileptic, or if a mother somehow suffers a upper respiratory tract infection during the first 3 months of pregnancy. So now that we have laid out the entire background of these diseases, remembering back to the Play-Doh demonstration, what do you think a potential solution for these disorders could be? Well, surgical methods. These are used actively today. Others are active management of environmental causes. And so where do I come in? I want to alter the genetic basis of these disorders such that their sufferers can develop in a normal and healthy manner. Human gene therapy already exists and is used in our healthcare system right now. This is my friend Max. Max passed away on May 3rd, 2021 from a lethal form of non-Hodgkin's B-cell lymphoma. Max received a state-of-the-art treatment known as CAR T-cell therapy. And essentially, CAR T-cell therapy works by extracting T cells, which are our body's immune cells, from a living patient's body and re-engineering them on a lab bench such that they function in a desired way and then are reinserted back into the patient that they came from. So what is the evidence? And how do we know gene therapy is a viable solution? Well, current research in the Albertson lab can speak to this. The environment can affect the development of bones, in particular, those involved in the jaw or feeding apparatus. We have shown in the Albertson lab that when bone is being deposited during development, there is a level of modulation that can occur based on the fish's immediate environment, and more specifically, how they are feeding in their immediate environment. We call this modulation plasticity. The bones are capable of changing how much bone matrix is deposited in order to best equip the fish's jaw for a particular foraging mode. There are genes directly responsible for this modulated or plastic bone response. And a lot of my work in the lab has been focused on the quantitation of plasticity. What genes and how many genes are responsible for it, and how they are regulated. The resulting data provide us a plethora of information, including where in the cyclid genome this plastic response is occurring. Believe it or not, you can do the same thing in the human genome. You can pinpoint where the molecular cause of a disorder is. Finally, developing an effective gene therapy to correct disorder is my goal. This will absolutely require more research to decide on many things. Are bone disorders caused by plastic genes in humans? During human development, what are the specific environmental interactions that could cause expression of plastic genes? We have reason to believe that it is because we see this in model organisms. And it ultimately comes down to genes responsible for cell proliferation and differentiation. Developing a gene therapy that is accredited or approved by many certified global organizations like the World Health Organization and the Center for Disease Control is our goal. And the cost for such a treatment is about $350,000 and above. With more research, it will only become more practical and affordable. Now, we must talk about the ethics of such a solution. With the wrong motive, gene therapies can have negative consequences, and we have seen this in other countries outside of the United States in the past several years. And I don't bring up these topics to deter you. Rather, it's more to inform and educate why such a great solution requires great responsibility. And so why hasn't this problem been solved before, you may ask? Why now? Why is now the time to solve this problem? And it's because there's limited research in this field, and human gene therapies are just becoming perfected enough so that their use is permitted on humans. But no matter what, it absolutely still requires a plethora of research. So, what can you do? As an audience member, support, advocacy, and awareness are all desired. Donations are excellent, but not at all required or requested. Donations would go to a grant fund that appropriates money for this research, specifically my research. My request to you as an audience is that you remain open-minded and to remember that there are so many benefits to be gained from such a solution. And if you have concerns, voice them. It is my job as a scientist to prove to people why my particular project, experiment, explanation, solution has validity. Thank you so much for listening.